Okay, thank you and welcome everybody to today's OICR Monday session about biomarker assays from development to clinical adoption. Okay, I have no relevant disclosures for today's talk. And so today's session, we really want to talk about how we're moving biomarkers from discovery into clinical adoption. And that's a topic that has become increasingly of interest to me and to a number of other people in this province and others. And we thought it would be best to focus it around a particular disease so we could highlight some of the issues. And we're going to be focused on acute leukemia today. So as Shola has already told us, we're going to hear from three speakers. So Michael Rauh is going to talk to us about nucleic acid biomarker assays in AML, talking a little bit about how we actually identify biomarkers in that discovery setting and why they're important and what the clinical utilities might be. Dr. Minden is then going to take over and talk a little bit more in the therapeutic area, targeting leukemia over time. And then we're going to close it out with Dr. Williams, who's going to talk about some of the barriers in terms of actually adopting biomarkers into cancer management. So in terms of the biomarkers that we're talking about, mostly we're going to be talking about genomic biomarkers, but of course what we say can be equally applied to any type of biomarker. And I also show you here a disease timeline and sort of thinking about what types of biomarkers we might be thinking when we want to bring them into the clinic to use for patients. And certainly everything from risk stratification biomarkers at the beginning all the way through to monitoring biomarkers for either recurrence or for surveillance are the types of things we're interested in. And this slide is really meant to highlight the fact that for the most part, with Ontario Health Cancer Care Ontario, we've been bringing through diagnostic biomarkers and predictive biomarkers because those are really the easiest ones for which the clinical utility is obvious. We're starting to understand more about bringing prognostic biomarkers into this space. We still don't have a lot of biomarkers in the surveillance and monitoring space. And in somatic disease, we don't really think so much about risk stratification biomarkers. So the types of biomarkers that we're bringing forward for clinical use tend to be things for which we have, for which there's a great deal of clinical utility. When we started to think in Cancer Care Ontario about how to start adopting biomarkers, and when I say adopting a biomarker, that means the Ministry of Health is now paying for it to be used for patient care, there was really no framework with which to do this. So, and Christine will be talking about this at the end, but we've really been working on trying to put together at least a loose framework that will help us to make consistent decisions in Ontario about when biomarkers could come forward and should be funded through the ministry. And that includes these steps, and they aren't really necessarily done in this order, but the steps include things like using NCCN or other guidelines in other jurisdictions to find biomarker compendia to identify what other jurisdictions say is useful for a particular disease. We spend a lot of time reviewing the evidence that would say that a particular biomarker is useful. We get a lot of volunteers and we use a lot of their time through expert working groups and doing this evidence review. We then try to categorize the indications for who should be tested, exactly which genes or other biomarkers should be tested. Then we look at the volumes and the cost estimates, and then the whole package goes to the Ministry of Health for yes or no in terms of funding. This is a fairly unwieldy process, but it's better than no process at all, which is what we used to have. And for the most part, the types of biomarkers that are being approved in Ontario right now fall into those top three categories, mostly predictive actually, because those are the easiest ones for which the clinical utility, which really refers to the impact of the biomarker on patient management, is quite obvious. Diagnostic biomarkers are also quite useful, and we're now beginning to understand that prognostic biomarkers, which just say something about the disease trajectory, can be useful as well for clinical management. Where we run into a lot of difficulties around the emerging prognostic biomarkers, and we don't have a great handle on how to stay up to date and make sure that the biomarkers that we're testing are actually the best ones we can do. In the solid tumor space, we've made a huge number of changes just in the last year, but in the hematologic space, we've actually also done quite a bit in Ontario. And this is just to show you that we actually had an acute leukemia provincial plan that rolled out in 2017. And you can read here that this plan was to provide an overview for how adult services are defined and should be organized and delivered. And this was, of course, in the days before CCO had anything to do with pediatric malignancy. That's changing now. But what you can see is that this provincial plan put forward a number of guidelines, the third one of which was this consensus pathology recommendations for complex malignant hematology. And in this document, and I just show you the front page of it here, in this document, we actually made recommendations about a number of biomarkers that should be tested, and mostly in the acute leukemia space. So this was actually a big win, and this was something we did fairly early on. But this is a very, very active area. And certainly in the acute leukemia in genomics, there's a huge amount going on. And so the problem we're faced with is really, how do we keep up? How do we make the decisions? And how do we make sure we're looking at the best biomarkers? So I want to just now take a minute and introduce the speakers. So Dr. Michael Rauh will be speaking next, and he's here, an associate professor in our Department of Pathology and Molecular Medicine at Queen's. He's right next door to me, actually. I'm not going to read through all of this, but Michael Rauh's research program has a strong focus in the myeloid diseases, and he's going to be talking sort of in that more discovery space. Then we're going to move on and talk with Dr. Mark Minden, and he's a professor emeritus in the Department of Medicine and Biophysics at U of T, but was also previously the head of the leukemia program at PMH, and he's going to be focusing more on the therapeutic biomarkers that we might want to think about in this context of acute leukemia. And then we're going to finish it with Dr. Christine Williams, who probably doesn't need an introduction here, but of course she's the executive VP for OICR, but also head of the implementation science group, science team. And that's really something that is starting to get a lot of legs and a lot of interest in the province and actually in the country. And so she's going to talk to us a little bit about what she sees as the barriers and maybe the potential solutions. So I'm going to stop here and ask Mike if you would like to take over and talk to us about nucleic acid biomarkers in AML.
Okay, thank you very much, Dr. Feilater. Thank you, Shola, Michelle, and Jai, and everyone at OICR for the opportunity to speak today. Try and advance the slides here. So I'll be talking about, there are many biomarkers in cancer and acute myeloid leukemia, but I'll focus today with the limited time on nucleic acid biomarkers. Previously, I entered an early access agreement for a myeloid NGS panel from Thermo Fisher, and I will be talking today about emerging technologies and research applications. So we're all on the same page about the disease. I'm going to provide an overview of how nucleic acid biomarkers play especially an important diagnostic and predictive role in acute myeloid leukemia, while discussing the standards involved with assay development and optimization and validation. I'll raise some of the challenges, which we'll build on a bit more with subsequent speakers. And I'll weave in two case studies, one of, if you will, one-stop shopping for sequencing, an all-in-one assay, and also a made in Toronto, made at OICR success story of a leukemic stem cell signature assay for patients with acute myeloid leukemia. On the left here, showing you a depiction of the hematopoietic hierarchy or blood cell production. And at the very top is the HSC or hematopoietic stem cell. And I'm just showing you the myeloid arm of hematopoiesis. Hematopoietic stem cells can also produce lymphoid cells like T cells and B cells. But when you hear of myeloid, acute myeloid leukemia, it refers to this arm. And acute myeloid leukemia arises when there are mutations in hematopoietic stem cells that lead to a block in their ability to mature. And what we subsequently see here downstream is that there are lower amounts of mature blood cells circulating, such as lower erythrocytes or red blood cells. People can feel tired, lower amounts of neutrophils and monocytes that would fight infection, and lower numbers of platelets that might help us to stop bleeding issues. And AML occurs when a hematopoietic stem cell acquires a mutation that leads to this expansion. Now, traditionally, I'm a pathologist. We traditionally diagnose these and quite easily can recognize these by looking at blood or bone marrow. And historically, the cutoff has been if we find at least 20% of cells in the blood or bone marrow that are these stem cells or blasts, they're also known as. But as Dr. Minden and other clinicians are dealing with now, we're realizing that on the horizon, on the very near future, there's more of a genetics first approach, and the actual blast percentage, which is really arbitrary, is becoming less important. And so we'll have to be especially recognizing genetic biomarkers much more early. This is just to walk through briefly as a pathologist, the samples that we get in the lab and how nucleic acid biomarkers fall into the diagnostic algorithm. So with informed consent, the patient would be draped and anesthetic would be provided. And bone marrow is obtained from the posterior iliac crest usually. The needle pierces through the cortical bone, and we can obtain withdraws several tubes that will go to various parts of the laboratory. So morphological features, we make slides and look at the actual cells under the microscope. Some cells go to the immunology or flow cytometry laboratory where we can quickly identify their nature and their lineage, so determine the blast type. And then tubes are collected for traditional metaphase karyotype cytogenetics and fish analyses, and more with molecular studies. Like with other tissues, we can also obtain a tissue biopsy as well, but these have less of a role in the case of acute myeloid leukemia. I'm going to focus on nucleic acid biomarkers. These nucleic acid changes or mutations are at the heart of AML. And in an oversimplified way to present this to you, there needs to be at least 2 hits that occur to get AML. The first is that differentiation block, or that block in maturation that I mentioned. And interestingly, most of the genetic aberrations that cause this, or the common ones, tend to be gene fusions that result from the translocation of parts of the chromosomes. And some examples are shown here. But that's not enough to cause acute leukemia. There needs to also be a hit that enhances the proliferation of this clone, and that typically occurs in genes encoding components of tyrosine kinase signaling pathways. And those 2 together can result in the acute leukemia phenotype. Historically, these biomarkers were measured using two main techniques, and that is more laborious, time-consuming metaphase cytogenetics in fish, and then targeting as more and more markers of this enhanced proliferation became known by individual molecular assays. However, our understanding of the nucleic acid biomarkers that are driving AML and predicting outcomes has been expanding. This is a diagram showing the breadth of our knowledge now. Probably the majority of the exonic mutations and gene fusions are known, if not all known at this point. And how they interact with each other is complex, and not everyone's leukemia is exactly the same. And so this is not becoming as conducive to single assays. What I'm showing you here as well is the upcoming 5th edition of the World Health Organization Blue Book classification, which is the standard around the world for acute myeloid leukemia. And more, as I mentioned earlier, we see in the red box here is a genetics first approach, where we're looking for these nucleic acid biomarkers that can lead to the specific disease phenotype in each patient, and less leukemia defined just by looking under the microscope. The issue is that with this exciting growing list, we need higher throughput and more comprehensive assays to find these biomarkers. And the traditional ways are finding their limits now. I'm going to focus mostly on sequencing-based assays, but there are several high throughput approaches to biomarker profiling. If these come up later in discussion or questions arise, these include multiplex ligation-dependent probe amplification, so you can look at a breadth of targets, copy number, or gene alterations. Optical genome mapping is quickly also providing technology that can replace the laborious traditional metaphase cytogenetics. Then comes sequencing technology, so we can look at a targeted approach at certain regions, certain genes. We can simultaneously also, using this technology, detect chromosomal copy number changes. Or rather than a targeted approach, some are leaning towards more of a whole genome and whole transcriptome-based sequencing to look for biomarkers and their breadth. And then finally, there are specific technologies that can be used that are really quite amenable to clinical practice, and I'll highlight this example of nanostring-based counting of nucleic acids. So a bit more about validation and how, if you're interested in validating a new biomarker, how to go about this. There are various guidelines that have come out in the last few years. This one at the top is a collaboration between the Association for Molecular Pathology and the College of American Pathologists. And on the bottom here is showing you standards from the American College of Medical Genetics. And these do overlap and complement each other. And some of the parameters that one would need to look at to validate a new assay would include sensitivity and specificity. So this relates to ruling in and ruling out diagnoses by looking for biomarkers, how well tests are reproducible and repeatable day after day, how likely are they to find the genetic aberration, and how likely are they to handle differences in samples and amounts of nucleic acids that are present and drawn from the patient, and how sensitive are they. These guidelines also cover really key issues, such as the importance of the reference material that is used to evaluate performance, guidelines about when you're looking for these, what sort of coverage of the genome is required, and how many samples should you start with when you're validating an assay, for example. We must also understand that the genome is not equal, and that there are regions that may be more difficult to cover depending on the nature of the nucleic acids there. And importantly, and we'll show you an example here as well, while you're validating, build in a process to look for clinical utility and to prove the utility of the assay and evaluate the clinical utility especially where resources are limited. And we also have to acknowledge, as Dr. Feileiter mentioned, that this is constantly evolving field and that we do have to think about updates to the guidelines as the knowledge increases. So we recently applied these, as I say here, to validate a targeted NGS assay. It's a semiconductor-based focused sequencing assay that looks at DNA hotspot driver genes of myeloid cancers, some of them requiring full coverage because, for example, tumor suppressors like TET2 have mutations that can occur throughout the coding sequence. And also now a two-in-one sort of assay looking at fusion drivers. And I mentioned their importance earlier in acute leukemia. Here there are 29 fusion drivers, and this assay covers around 700 possible fusion partners and combinations. We recently published this in the Journal of Molecular Diagnosis. I highlighted as well as a thank you to the OICR and the Ontario Molecular Pathology Research Network, who funded our Path to My Diagnosis study. And this was some work conducted by PhD student Christina Ferrone. So in the period over about a year and a half in 2018 to 2019, and in conjunction with Dr. Feileiter, we sequenced 187 nucleic acid samples from 168 patients who presented to our Health Sciences Center here in Kingston with suspected or known myeloid cancers. As Dr. Feileiter alluded to earlier, we did have funding for acute leukemia cases provided by Ontario Health and Cancer Care Ontario. These initiated the sequencing runs, but thanks to Dr. Feileiter and the laboratory, we're also able to help fill these runs. We don't see enough acute leukemias in Kingston with non-leukemic cases where we're screening and indicating there's a need. Some of our validation metrics, I'll just really briefly mention this, was that a 5% limit of detection for variants that are single nucleotide level with really high sensitivity and specificity. This assay didn't perform as well for small insertions and deletions, and our limit of detection was only around 10% of alleles for those. We tracked the clinical utility as was recommended, and in this highly selected sample, about 77% met our criteria for clinical utility or actionability. I'm showing you a summary of this here. The categories of utility were mentioned earlier by Dr. Feileiter. Most commonly, these help us to clarify or achieve a diagnosis by the World Health Organization Blue Book, and remember that list is now expanding. These variants help us to identify and risk stratify patients and lead to management decisions. So for example, they may help to identify a targetable variant for which there's a drug or clinical trial, or the likelihood of a patient benefiting from an allogeneic or unrelated bone marrow stem cell transplant. And so at our institution, this assay was highly clinically useful. Now, we did not, however, at this point yet, even though we're looking at RNA, replace traditional metaphase cytogenetics. That's something that we're looking at down the horizon, given the high concordance and ability of this assay to pick up those variants. But this paper that I wanted to highlight in the New England Journal of Medicine is from the Washington University School of Medicine Genome Center. And here they took a more comprehensive one-stop approach where they were trying to replace the traditional cytogenetic methods with an all genome sequencing based approach, and with high throughput and high quick turnaround. And so tumor cells are obtained from the patient, such as AML, DNA is prepared, libraries are constructed, and they sequence the whole genomes at about 60-fold average depth. And built into this, importantly, is the analysis, the validated analysis and reporting. And I just want to talk to you now about a little bit about some of the metrics and key findings of this paper. So they were aware of 40 recurrent translocations and 91 copy number alterations in a test set. All of these were detected by their whole genome sequencing approach. And in addition, on top of that, they found in 17% of patients additional genomic events that weren't identified by the traditional means. And they validated this in a prospective cohort where they turned this around at the median of 5 days and as early as 3 days if required. And again, finding lots of new genetic information. And so, the main barrier, someone's microphone's on. The main barrier was cost, however. They cited a cost of about $1,900 US per patient, but given the comprehensiveness of the assay and how costs are coming down so quickly, they figure that this may be something for others to consider. Now I'm going to briefly shift from DNA to RNA biomarkers. We may be able to uncover beyond gene fusions, transcriptomic changes, or other changes at the RNA level. And I wanted to highlight this Made in Toronto, an OICR study by Doctors Jean Wang and John Dick, and Dr. Minden was a co-author, and Stanley Ng was the lead author. And what they did in this case was to look for, with their interest in leukemic stem cells, a gene signature that might predict how adverse is the type of leukemia that the patient has. So the authors at OICR in Toronto and at UHN fractionated various populations of cells within AML patients, and then compared gene expression within these populations to identify a core set of 17 genes that predicted stem cell properties. And as this score increased, what they found was people are less likely to respond to AML treatment, they were more likely to relapse, and were less likely to survive. Now, this assay initially was based more on microarray data or transcriptomic data, which hasn't really found a great home yet in the clinical setting. But the authors did then start to apply this to a more clinically amenable platform called a nanostring encounter, which is a way to measure native nucleic acids like RNA without any amplification. And it has had great success in other cancers, especially Hodgkin lymphoma and diffuse large B-cell lymphoma, having very reproducible results. And so this paper that I'm highlighting here, if you're interested, is how they've moved towards clinical CLIA certification, OLA certification of this assay. And so this assay includes the 17 genes and 12 housekeeping genes. They can turn it around in 2 days. Looking at some of the performance metrics and assay validation, there is really low within run and between run variabilities. The results are quite consistent. It's a really user-friendly. What they do is they enable a normalized weighted score to be produced. So if we look over on the right side now, you can compare the patient's score for the LS17 to a reference data set and synthetic controls. And the cut point is right at 50%. So those that fall on this side with a score greater than 0.5 have increased risk and worse overall survival. So important for clinical assay biomarker validation. They applied this to a variety of sample types, fractionated versus unfractionated, different collection methods of blood tubes, and the assay was very robust. And importantly, also tried this assay, same assay at a different site, getting the exact same results. So there's independence of site, high technical reproducibility, which are all features that are really important for biomarker assays. So this is my last slide, and maybe something we can bring back again when we get close to the question time. Thinking of challenges and future directions, the big choice is going to be whether or not to focus on targeted assays, which are now a bit cheaper and easier to manage, versus genome-wide. And this is going to depend on local resources and expertise, cost, time, and of course, the clinical utility of the content. But generally, the field is moving from targeted to more comprehensive assays. This requires foresight and advocacy, and I participate on some of those panels that Dr. Feilater's mentioned. We try to publish, we try to advocate for patients and for funding and anticipate the needs that are coming down the pipeline. And then thinking about the sample type, I mentioned bone marrow analysis is standard, but that's really quite invasive. The patient has to be, as I mentioned, like draped and consented and having their marrow poked. But down the road, we consider more evidence for peripheral blood cells or even cell-free DNA in plasma. And really now, we didn't have time to get into, but we're just looking at the bulk of disease as the patient presents. But we know now that cancer specimens are quite heterogeneous. And as people are starting to look at the single cell level, that's providing another level of information that's really important, especially as we monitor patients' responses to therapy and the progression of disease. We need to really be thinking at single cell and residual disease levels. And I understand that Dr. Minden is going to be covering some of that. So I just want to thank you again for your time, and I'll pass over now to Dr. Minden.
Thank you, Michael. I'd like to thank the organizers for the invitation. I may go over time, and I thank the organizers also for a platform to talk about some important concepts about how we should think about the testing. And how do I move this thing forward? Okay. So my focus will be on the LSC17 signature, IDH inhibitors, and residual disease opportunities. Okay, so Basurga was an editor for Cancer Research, and he had 3 caveats that talked about how to approach cell lines or cells. And his caveats were applied to these cells under these conditions at this time, and shows the uniqueness of a cell, how you grow it, how you test it. And time has the component of when you test it, the techniques available at the time, and the treatments. I've transformed this to patients, and it applies to a particular patient or group of patients with the same mutations, treated with a particular therapy. And that's very important as our treatment of AML moves forward. And at this time, again, relates to how we test for the disease, how we monitor it, what supportive measures we have, and what sort of thinking we have. For example, age has been an area that's been debated and is evolving over time.
Mark, in the bottom left corner, there should be some very opaque icons for changing slides.
Oh, okay. Okay, so we've already heard the word prognostic factor. And so there are two different factors that we consider in the treatment of the disease. Prognostic factor, I see, just now. And we talk about disease being either good or risk or poor risk. In fact, AML is poor risk unless you have treatment for it. And so one has to, when you talk about a prognostic factor, describe what your treatment approach is. We also have predictive factors, those that will tell us more or less whether or not a patient will respond to a particular treatment. And this really gets down to certain types of mutations, and they can be used either at presentation or in post-therapy. The LSE17 signature was derived from variation in gene expression of 4 subsets of cells from AML patients based on CD34 and CD38 and their ability to engraft. The gene expression was assessed by a microarray technology, and 17 genes were identified that reflected differences in leukemic stem cells. Some of the markers were increased expression, others were decreased expression, and they identified patients with a median better or worse survival. It's of note and important that in this study, all patients were treated with daunorubicin and cytarabine, usually 60 milligrams of daunorubicin and then varying dose of cytarabine. And these are the 17 genes. And you can see that each one has a different weight to it. Next slide, please. Idea of this signature was to predict who was going to benefit from a 3 and 7 type regimen. And you can see that there were patients who had low signature who did better than the patients with a high signature, but it's still not great. You're not getting cures with this. And I think that's one of the criticisms I have. One would like to be even more precise than this allows. And this may be a problem of using median cuts and so on. Next slide, please. So if you look at the back one, if you look at the actual genes in the signature, 34 shows up because that's how you selected the cell. So it had to be there. But what are other markers telling you? And one of the ones that we've looked at a little bit is SOX2. And SOX2 high expression is probably a mark of the presence of a FLAT3 ITD, because SOX2 is actually a protein that leads to dephosphorylation of signaling pathways. The other thing that SOX2 tells you is that SOX2 antisense 1 is being expressed, and that in fact is a protein that protects the cells against cell death. And I think as we look into the other markers there, they have a role in leukemia that is important to understand more as a potential target rather than as a part of a signature. Next slide, please. And so do we make of the LSE17? and how to adopt it. Well, the problem with adopting it is there are many other signatures out there that are giving about the same sort of information. And the uniqueness of the LSD17 is that it started with leukemic stem cells, but the other signatures that are coming out have about the same split. There are other considerations too that go into deciding about how to treat somebody. Their age, premorbid conditions, mutations they may have that may override the signature. The treatment landscape is changing. This signature was based on a 3 and 7. We're now doing 3 and 7 plus Mylotarg. We're now doing venetoclax azacitidine for patients who are over the age of 60, and it's creeping down below the age of 60. And we don't know how the LSD17 works in that setting. And then the other thing it does is it doesn't tell you how to treat people. It tells you who shouldn't get 3 and 7, but doesn't say what they should be getting in addition. So I think that these sort of signatures really have to be thought about as to how we're ever going to use them, especially as treatments keep changing. IDH is recurrently mutated in AML in 10 to 20% of cases. It is occasionally seen as a pre-leukemic marker. IDH1 is mutated at 132 and IDH2 at Arginine 172 or 140. The IDH1 is cytoplasmic and IDH2 is mitochondrial. IDH uses glutamine to produce alpha-ketoglutarate, but with the mutation, you get 2-hydroxy glutarate and DNA hypermethylation that then changes the behavior of the cell. The mutations are not likely equivalent, as, for example, IDH2 is not seen with NPM1, and with IDH inhibitors, you actually see different levels of response depending on mutation. IDH per se is not an oncogene. The mice who have a mutant IDH don't get a leukemia. They may show a shift in their phenotype, but they don't have overt high count disease unless you cross them with something like a FLT3 ITD mutant. There are a number of treatments coming along, but one of the first places where we saw that IDH made a difference was when it was combined, when the treatment of IDH mutant AML was done with venetoclax and azacitidine. And there, rather than the usual sort of 40% response rate with, or 40 or 60, depending on how you measure response, with just azacitidine, the Ven-Aza had a very high response rate of 79% and a prolonged survival of 24.5 months versus 12.3 months for Ven-Aza in non-IDH cases. So there's something different about these patients. Okay, so the response to IDH inhibitors, there are now 2 out there, one against IDH1 and another against IDH2, and then a third one, there were one against the combination anacetinib against IDH1 was tested in by itself at 100 milligrams a day for 28 days. And there was a 20% response rate with the median time to response being very quick at 1.9 months. Patients with the 172 mutation did somewhat better than the 140 mutation, and the median duration of response was 5.8 months. It's important to note that even with treatment, the mutant allele still persists, even though at a flow level, you don't see evidence of the disease, or by microscope, you don't see a lot of blasts. So how you are detecting residual disease is important. We'll come to that. Patients who had a lot of mutations had a low response rate. And so if the patients had more than 6 mutations, giving them the anacetinib wasn't of much value. I made a mistake. Anacetinib's for the IDH2. Ivosidenib for IDH1, 500 milligrams every 28 days. They had a response rate of 41% with 22% complete remissions, time to response 2.7 months, and median duration of 8.2 months. The combination of IDH inhibitors with azacitidine or azacitidine with venetoclax are very active, and long remissions in excess of 20 months are being seen. So we're going to be seeing that the IDH inhibitors will be used either with AZA or AZA and Ven in the future. And the combination with AZA, or the fact that the IDH mutant cases have a good response with venetoclax, is likely due to the fact that they have a high hypermethylation rate. And so the azacitidine or decitabine is reversing that. And the venetoclax is targeting BCL2, which is important as a survival factor in the IDH mutant AMLs. Next slide. There are resistances to IDH inhibitors, and those are NRAS, SRSF2, PTPK11, FLAT3 ITD. You can see mutation of the other IDH isoforms. So if you treat with an IDH1 inhibitor, you can see an IDH2 inhibitor come up. TET2 mutations can also cause resistance because they too cause change in methylation. Presence or acquisition of a RUNX1 mutation or acquisition of the CBP alpha mutation are also associated with resistance. Next slide, please. So the approach to AML therapy is no treatment, supportive care with transfusion, or curative intent therapy. And that is typically induction chemotherapy with the goal of remission, and that's followed in some cases by allogeneic stem cell transplant. And then we stop treatment. We're like the three monkeys. And this has been very dissatisfying for most of my career, that we don't do anything other, you do a transplant and then you stop. There came along the concept of MRD, or minimal residual disease. This was first used in a surgical setting where a tumor was cut out and the surgeon thought they had cured the tumor in the mouse, and then the tumor came back. And this gave rise to the post-surgical therapy. In the context of the leukemias, MRD is not minimal residual disease. The minimal residual disease actually probably occurs about day 10 after chemotherapy. And by day 28, it's probably regrowing already. And minimal doesn't have the concept of time to it that is important in our management of acute leukemia. People have talked about measurable residual disease, but that's an oxymoron. If you're testing for it, you're measuring it, you're looking for residual disease. People have then said MRD is molecular residual disease, but there are other ways of detecting it. And so my preference is residual disease monitoring, or RDM. This involves looking for disease over time after therapy. And it can either be visual inspection, which Michael has already indicated isn't very good, flow cytometry that can pick up anywhere from 1 in 100 to 1 in 1,000 residual leukemic cells. And then you can use RNA expression or the detection of fusion transcripts, DNA detection of mutations or methylation signatures, or protein expression to start to get to detecting 1 in 10 to the 4 and 1 in 10 to the 5 residual leukemic cells in the patient. For testing, you can either use the bone marrow or peripheral blood. And as Michael mentioned, we're moving more towards plasma and serum because they sample the whole body, and there may be sampling errors in using just the bone marrow. In reporting RDM, I think there are 4 critical points. One is the source of the sample, because there may be bias if you're doing a marrow and you're not where there's disease, you're going to miss it. The time of the sampling in relationship to treatment, the method of detection has to be defined, because as I said, flow has a very different level of detection than the molecular methods. And it's also important, I think, to talk about the lower limit of detection of the method being used. And so we now can start to look at other ways of assessing disease and making a difference for the patient. We can now determine the presence of disease across time. And the exemplar of this in leukemia is CML, where, or Philadelphia positive ALL, where we look at the Philadelphia chromosome BCR-ABL at presentation, a couple of months after starting imatinib, 6 months, a year, and so on. And we do this with a Philadelphia positive ALL as well. We can start to identify patients who are cured, and there's no evidence of disease at what's the level, what's the method, and may not need a therapy. And recently there was a publication showing that in colon cancer, this is in fact the case. There are patients who don't need any sort of therapy after resection. We can use measuring the level of disease as how to monitor the effectiveness of maintenance therapy or giving more therapy to get a deeper remission. A deeper remission is important for outcome of transplant. Patients who go into transplant with evidence of residual disease often relapse, whereas they go in without evidence of residual disease may have a long-term outcome. And we can use detection of residual disease to alter our therapy. I mentioned transplant as a way of eliminating residual disease. This really is the Hail Mary. You'd like to get rid of it. It causes lots of other problems. And the patient who really needs a transplant is one where you don't have normal cells coming back. But we may be able to change how we're treating patients by adding p53 inhibitors or demethylating agents. And there are immunotherapy approaches coming along. There are cellular approaches next, such as double negative T cells, NK cells, CAR T cells, cytokine expressing cells, checkpoint inhibitors, bispecific antibodies against CD33, CD123, and transcription modulators. For example, we now recognize that ATRA may be of value in upregulating HLA-DR and HLA-DR negative forms of AML, and so enhance immune recognition. And so the bottom line of residual disease monitoring is, once you've achieved normal hematopoiesis, there are multiple ways to eliminate or control residual disease. And I think that's the exciting part of the next decade in the treatment of leukemia. Thank you.
Hi, everyone. So thank you both. Really, really interesting talks. And I feel humbled to be following them. I'm gonna have the same problem as everybody else with the screen controls. Okay, so I think everybody's sort of familiar with this kind of schema, and OICR was really created to address the challenges that Mark and Michael just talked about in moving research between really the early discovery, basic biology findings into that preclinical setting, and then clinical research, later translational research, and then into adoption in the health system. And it's obviously not nearly as linear as this handy schematic shows. And as you just heard, there's lots of back and forth between these different steps, but both gave great examples of the way we can move science into clinical care, or try to move it closer to clinical care. So I'm increasingly interested in these later stages in overcoming some of these gaps, particularly moving research from clinical to late translational research, and then particularly the phase of adopting it in the health system. So I'm kind of interested in last mile problems, I guess, is another way to put this. And I was really humbled by this revelation back when I was at the Canadian Cancer Society, when I was in charge of the research program. And, a volunteer told me, research alone can't cure cancer. And that's absolutely true. So we need all of these other things. We need clinicians to use that research. We need patients to want the changes we're proposing. We need health systems and hospitals and caregivers that are able to deliver it. We often need industries to produce it at scale. And we certainly need governments in our health system that will pay for it. And underlying all of that is we need to know whether that research really works. So in recognition of this, recently, in the last year or so, we've put an increasing emphasis at OICR on implementation science. And we're still really defining what this program is going to look like, but it really recognizes, I think, what Michael put very nicely, which is that we sometimes need foresight and advocacy to move research into use. And I think there's lots of foresight. Many, everybody on this call shares it. Probably what we need to do a little bit more of is the advocacy part. So implementation science, I like this definition from the University of Washington. It's really simple. And it's really that idea of facilitating the uptake of evidence-based practice and research into regular use by practitioners and policymakers, in some cases, payers. In our case, in Ontario, we have partnerships all over the place, and OICR is all about collaboration and building those partnerships. But at the end of the day, there's a very key partnership we all have with Ontario Health. Previously, it was more closely with Cancer Care Ontario, but as you know, Cancer Care Ontario is now part of Ontario Health, so we have a single health agency. And as we do translational research and support it at OICR, we're looking for the pickup on the other side of that from Ontario Health. And that is sometimes through the Ministry of Health as well. So this partnership is really critical. You'll see that on this slide, I called it a learning partnership. Maybe that's optimistic, but that's where we want to get to really, is that sort of learning health system. So this slide just shows, there are different ways to define this, but Harriet and I have tried to set up what are the phases of oncology biomarker development and adoption. And these are just the basic building blocks of biomarker X, in which the really we hope it to be driven by patient need, but there's the discovery and biological validation of a biomarker in that sort of preclinical and clinical research setting. And then we show that there's clinical utility and validity to that biomarker. We have to establish technical parameters of how to do that assay. That assay has to be adopted more broadly by clinical labs. It needs funding. There's clinical lab validation. So in the individual labs delivering that assay, we need to see that on-site validation. That can be in hospital or an industry setting. And then there has to be ongoing proficiency testing to make sure that assay works. And as part of that, we want to see scale and spread of that biomarker into our care systems. You'll notice that there's a big red circle on this slide, and this really is where we see roadblocks. So where there's really a gap in this pathway. There are gaps, obviously, in various parts all along here, but really here is where ownership of this development and adoption pathway sort of falls to the wayside. And you'll notice that it's really clustered around that very critical point in the Ontario system of Ministry of Health approval. So barriers to adoption, to go into a little bit more detail about those three phases. So clinical utility and validity. How do we know that this biomarker or any innovation works and works consistently? What evidence do we need to determine its clinical utility? Is it cost effective? And what is required for the ministry to approve it? So what does the ministry look for in a package of evidence? And Harriet referred to this at the beginning of her talk as well. The next stage around technical parameters, which assay should be used to test for this biomarker? Often there are multiple assays available. How do you know which one to use? How do you know how to do it standardly? What are the limits of detection, measurement? How reproducible is it? And again, who's going to pay to do this workup of this test? And then the next stage in that sort of middle of roadblock is adoption and funding. And funding, of course, is key. How are the best practices for the new biomarker shared amongst labs? Who's going to pay for the test? Who's eligible for it? So what's the patient selection criteria? And then how do we make sure that this actually works in the real world, so outside of a clinical trial setting? So this is where we're really working on these sort of gnarly problems. And some of our proposed solutions are what I'll talk to at a really high level shortly, particularly focusing on this idea of an innovation pathway. I want to convince you of the need for evidence checklists. I'll talk a little bit about an implementation lab that Harriet and I and others are piloting, and then make a pitch for more money for real world evidence studies, which of course often depend on data sharing, and data sharing in particular between the clinical health system and research. I'm not going to get into that in this talk. It's a whole other subject. But certainly a critical enabler of us being much more effective at actually having a learning health system and actually learning from each patient. Okay, so I want to talk first about the innovation pathway and convince you that, first of all, we need one. Harriet talked about this at the beginning, but there really is no structured way at looking at any new innovations coming into the health system and whether or not they should be adopted, should be paid for, and how that should happen. So what we are proposing on the surface is something pretty simple. And that is in order to bring more innovation into cancer care, we need some sort of multi-step, structured, and importantly, transparent framework to guide evaluation and adoption of new technologies and processes. So what do we mean by all that? This is based on work that was a huge consultation a couple of years ago, just pre-pandemic, with a lot of different viewpoints. Some of the ones I talked about in that slide that talked about research alone can't cure cancer. So all of those types of stakeholders. And there's a larger document, quite a long white paper on our website if you're interested in more details about this, that came out of a significant workshop we had. But I just want to summarize a couple things from that work. And the first is the guiding principles and considerations that we think are critical. And when I mean we, I mean all these different stakeholders in a decision-making framework. So first of all, we need a more nimble system. I don't think anyone's going to argue with that. Technology development is iterative, and we need a system that can adapt to new technologies and new findings, and it needs to be faster. We need a framework that is broadly applicable to all kinds of different technologies, whether they were developed in Ontario or coming from global innovations. We need to really embrace this concept of a bias to permissive. So rather than saying no to new technologies and all those roadblocks we put in place, we need to be more open to more small tests of innovations and much quicker pass-fail decisions, and that's really critical to allow that permissiveness. We need a system that's transparent, whether you're coming from an academic background or you're coming from industry. New tests, we need a clear entry point for decisions at the ministry. It needs to be open governance so that we know how it's governed and not privilege access to any one group. We want to leverage existing organizations. So putting a framework in place doesn't mean creating a whole bunch of new things, but actually integrating some of what we have already, particularly around health technology assessment. It's critical that there be discontinuation and disinvestment. Again, many people have worked in this type of area and pushing disinvestment in things that really don't work. And some of you may have heard of the Choosing Wisely initiative, for example. But this requires collection of evidence. And if there is not evidence of benefit, we need to discontinue doing things that aren't effective. And then finally, we want to build all this within a learning health system where there's really a feedback loop and, as I say, better data sharing between research, patient experience, and decision-making. So this is just in one slide, a summary of what we've proposed go into this innovation framework. That it has 3 phases. The first being a really appraisal phase where we're determining whether the innovation is a priority, clinical utility, health system readiness. So essentially a prioritization at the beginning to say, is this a technology we can even consider, and is it something we need? The middle phase is where the heavy technology assessment happens, and this is where we need to determine things like clinical validity and the cost. And then the last phase is around implementation. and knowing whether it's cost effective, and again, did it perform as expected. So this is the sort of framework we've proposed to the ministry that's really needed, not just for cancer care, but for healthcare in general. But we're really focused for now on cancer care. So status and ongoing work with this. I mentioned the OICR white paper that was published in 2019 and is on our website. We have had, I guess the good news is we have had, especially recently coming out of the pandemic, much more buy-in from the Ministry of Health about this. The genetics branch in particular really sees the need for this because so much technology change is coming in the field of biomarker development, genetics, genomics. I've also heard that Ontario Health and CCO have actually assigned staff to developing this framework. We know that there was a separate report done about the need for it. And at the moment that there's work going on at Ontario Health to sort out the governance and ownership of this sort of framework. As I say, genetic testing for cancer is the immediate priority, and groups like the new Provincial Genetics Advisory Committee should support this. So it is pushing a rock up a hill some days, but this is where the advocacy part really comes in, where we're really working with many stakeholders and partners at Ontario Health and CCO in trying to move this forward. The other piece that we're trying to do on the side and in a smaller collaboration is OICR is collaborating with CADIS, so the Federal Drug and Technology Assessment Agency, and also CPAC is involved, to come up with a piece that's needed for looking at biomarkers and making decisions about them. And that's the idea of developing an evidence framework. So what kind of evidence do we need to assess genetic tests, especially biomarkers, and come up with consistent evidence standards to support decision making? So in other words, if you are trying to get a biomarker into the health system, what evidence do you need to show that this works? How do we know that it works? And what we'd like to come out with from this roundtable is actually a checklist, so that you know whether you are the investigator trying to push a biomarker forward into clinical care, or you're an industry, or your government, what are the rules about what we want and what levels of evidence do we need to know is effective. One slide on the implementation lab. So this really addresses that gap around standardization and quality control. And the idea, this is an idea that was, I really have to give credit to Harriet for, funded by OICR, but based at Queen's through Harriet's lab. And a collaboration with multiple other groups, including diagnostic development, some pharmaceutical companies, and UHN. And this is the idea of having a centralized place in which we can generate quality and performance assessment metrics for biomarkers, establish how to standardly do the assessments, report on the comparative analyses, and then share that information with other hospital labs, clinical labs. Where this pilot study is now funded, where we're looking at lung adenocarcinoma biomarker testing. So we're at the phase of collecting specimens and doing the workup. And we're hoping to expand this concept with more funding from the Canadian Cancer Society. And we can always address that at the end. Harry can answer questions too. And then I wanted to also add a pitch for another thing OICR is doing around implementation science, which is funding innovation to implementation grants. And we've had one round of these proposals that were really supplements to existing OICR projects. We now have a new round of applications available, RFAs out. And you can see there the purpose of the funding, really supporting evidence needed to overcome implementation bottlenecks and or knowledge synthesis, dissemination, exchange. So these are, we've doubled the amount of money available. That's the good news if you looked at this before. They're now $100K per year over 2 years. There'll be an information session coming up about this in September, and it's an LOI deadline. So it's a 2-stage application. The LOI deadline's in October. So coming back to this slide, and this is where I'll end. This is how we're trying to fill some of the holes in this development pathway. So around clinical utility and validity, trying to establish standard evidence checklists. And I didn't mention this earlier, but we're trying to do this nationally rather than province by province, so that how you assess and determine whether a biomarker is effective is the same in Alberta as it is in Ontario. And we don't reproduce each other's work every single time in every single province. The technical parameters, we're hoping to help seed that work through the implementation lab, and then adoption and funding will be done through information sharing, again funded at the moment through research grants through OICR and CCS. And importantly, that decision point around the Ministry of Health approval and funding, we really hope to be much more nimble, much more transparent, much more effective with the adoption of a decision-making framework there. So the outcomes, I think, are obvious. We need more innovation in our health system. We want to see more active learning, and all of us want to see better outcomes for patients. Although I've only just touched on it in this talk, the other interest, of course, is allowing for more commercialization and more IP to be established in Canada and to make that easier in Ontario. And also to encourage more private sector collaborations and investment. And sometimes that's controversial, but I really feel that in order for us to be effective in getting more innovation into the health system, we need to work with the private sector as a partner. So I'll end there and hand it back to Harriet.